Welcome to Cell Biology. Today, we are going to explore one of the most brilliant and essential mechanisms in all of biology, the chemiosmotic hypothesis. This is the fundamental scientific process that explains exactly how your cells generate the energy required to keep you alive, breathing, and thinking. Whether you are a completely new student to this topic or just looking to deepen your understanding, by the end of this lecture, you will know exactly how your microscopic cellular engines work. To understand this hypothesis, imagine a massive hydroelectric dam holding back millions of gallons of water. The water naturally wants to flow down, but the dam blocks it. When the gates open, water rushes through, spinning giant turbines to generate electricity. In your cells, a very similar process happens. Instead of water, your cells pump tiny charged particles called protons across a barrier, building up a massive biological pressure. When these protons flow back through a special cellular turbine, they generate ATP, the energy currency of your body. Now that we have a mental picture, let us look at the formal scientific definition. Proposed by the brilliant scientist Peter Mitchell in 1961, the chemiosmotic hypothesis explains how cellular respiration produces ATP. The energy released from the electron transport chain is used to pump protons across the inner mitochondrial membrane. This pumping action creates a powerful electrochemical gradient. This gradient has two parts, a chemical difference in pH and an electrical difference in charge. Because nature hates imbalance, these protons desperately want to flow back to equalize the pressure. This immense pressure brings us to the mechanism of energy creation. The mitochondrial membrane is strictly impermeable to these protons, meaning they can't just slip back through. Their only way back is through a specialized enzyme complex called ATP synthase. Think of this as our cellular turbine. As the protons physically flow through ATP synthase, the enzyme actually rotates. This mechanical rotation physically forces ADP and inorganic phosphate together to synthesize ATP. It is a literal microscopic motor spinning inside you right now. So why does this specific mechanism matter so much to you and me? Simply put, this chemiosmotic mechanism is responsible for generating over 90% of the ATP your body needs to function. Every heartbeat and every thought spends ATP generated by this exact proton gradient. This is also why certain poisons are so deadly. If a toxic chemical, like cyanide or carbon monoxide, disrupts the electron transport chain, the proton gradient collapses immediately. Without the gradient, ATP synthase stops spinning, ATP production halts, and the cell dies within minutes from energy starvation. Before we move on to the mathematics, let us see this with a concrete visual. Here we are looking at the inner mitochondrial membrane. Notice the top area, the intermembrane space? It is packed with positively charged protons, creating a highly acidic, positively charged environment. Below the membrane is the matrix, which has very few protons. The membrane itself acts as the dam. On the far right, you can see the ATP synthase enzyme. The protons flow down through this channel, and as they do, the bottom portion of the enzyme rotates, snapping ADP and phosphate together to form ATP. Now that we understand what the gradient is, let us look at how we measure its power. Biologists call this the proton motive force. This beautiful equation, derived from thermodynamics, combines both the electrical pull and the chemical pull acting on the protons. Delta P is the total driving force in millivolts. Delta psi represents the electrical voltage pulling the protons. The second term represents the chemical concentration pushing them, which depends on temperature, T, and the difference in acidity, delta pH. If the voltage difference increases, or if the outside becomes much more acidic, the total driving force increases dramatically. Let us put this concept into practice with a realistic biological scenario. Imagine a healthy mitochondrion inside a human muscle cell. At human body temperature, the complex constants in our formula evaluate to roughly 60 millivolts, giving us a simplified equation. We are given an electrical gradient of 160 millivolts and a pH difference of 1 unit. We plug these values into our simplified formula. Multiplying 60 by 1 gives us 60 millivolts for the chemical pressure. Adding this to the 160 millivolts of electrical pressure gives us a final answer of 220 millivolts. This massive force is what violently spins the ATP synthase turbine. This formula we just used leads us to an important question. How much actual energy does this voltage represent? To find out, we use the thermodynamic equation for Gibbs free energy change, denoted by delta G. This formula connects voltage to free energy. The variable N is the number of moles of protons. F is the Faraday constant, which is a fixed value of 96,485 joules per volt per mole. And delta P is our proton motive force, but it must be converted to volts. If the proton motive force increases, the amount of energy released increases proportionally. Before we move on, let us see this with a concrete example using our previous result. We want to find the total energy released for 1 mole of protons, so N equals 1. First, we must convert our proton motive force of 220 millivolts into volts, which gives us 0.22 volts. Next, we substitute our values into the equation. -1 times the Faraday constant times 0.22. Multiplying these together gives us a final answer of -21,226.7 joules per mole. This tells us exactly how much energy is released into the cell. So we have calculated the free energy, but what does this number actually prove? Let us break down the physics of our result. We found that delta G is approximately -21.2 kilojoules per mole. The negative sign in this formula is incredibly important. In thermodynamics, a negative delta G indicates that energy is being released by the system into its surroundings. This makes perfect sense intuitively because the protons are flowing naturally down their gradient from high pressure to low pressure, releasing stored energy, just like water rushing out of a dam. This brings us to the final biological proof. We have established that every mole of protons rushing through the ATP synthase turbine releases 21.2 kilojoules of usable energy. The ATP synthase enzyme is designed to capture this massive burst of released energy. It uses this exact energy to physically snap a phosphate group onto ADP, creating ATP. By calculating this thermodynamic value, we have proven mathematically that the proton gradient holds more than enough energy to drive ATP synthesis and sustain all cellular life. To wrap up our journey, let us review the key takeaways. The chemiosmotic hypothesis is a beautiful, unifying theory of cellular energy. Remember, your cells do not make ATP directly from the food you eat. Instead, they use the energy from food to pump protons across a membrane, creating a powerful biological battery. We saw how the proton motive force measures this combined electrical and chemical pressure. Finally, when these protons flow back through the ATP synthase enzyme, that stored energy is converted into the mechanical rotation that synthesizes the ATP, keeping you alive. You now understand the fundamental engine of life.